Hi, I'm James Verdier, and welcome to the American Institute of Biological Sciences BioScience Talks, which is a forum for integrating the life sciences. On the second Wednesday of each month, and sometimes in between, we discuss the latest bioscience publications. And as a reminder, if you'd like to read more, point your browser to academic.oup.com/bioscience. Today's episode is a very special one. Late last November, I once again had the chance to attend the American Society for Gravitational and Space Research's annual meeting, which was held this year in Denver, Colorado. Now, this meeting is always a very exciting one, and there's obviously some natural energy that comes into play whenever you're talking about something as cool as spaceflight. But what always really strikes me is the amount of cross-disciplinary discussion and diverse collaboration that goes on. This is one of the few places where you can go and, in the space of a few minutes, meet plant biologists, fluid physicists, materials scientists, artists, students, medical researchers, and so on. I encourage everyone within the range of my voice to plan on attending their meeting this year in Houston between the 4th and 7th of November. But for right now, let's go to some of those great conversations that I had in Denver. First up is Dr. Kevin Sato, who is ASGSR's immediate past president, although he was president at the time we talked. Let's go to that interview. Dr. Sato, thank you very much for joining me today.
Oh, it's great to be here.
Okay, yeah, it is always great to be at the ASGSR meeting.
Yeah, no, it's one of these really nice, great meetings where we get an opportunity to bring specifically our science community who is interested in doing life sciences, physical scientists, and fundamental physics research all together in one place to really learn about the science that is going on in each field, to build and develop networks, see where we have crosstalk and cross-pollination of scientific research. But really, it's a really nice area to celebrate the unique type of research that we're doing, which actually is leading not only to understanding how life and physics and physical sciences work in the space environment, but also how it is helping to bring about the revolution in terms of commercialization of space and eventually bringing us back to the moon and then beyond to Mars.
Okay, and why don't we talk about the commercialization of space a little bit? What role does that play for ASGSR?
It plays a very large role. There are several areas of commercialization that we use. For example, to conduct our experiments in space. We have commercial groups who actually help to implement our experiments in space and operationally take us forward. We use commercial transport carriers. There are also new platforms that are coming about, such as Blue Origin suborbitals or other types of platforms that are really managed and run by commercial, that without them we would not be able to do our experiments in space. The other part too that's important is the ISS National Lab. And what that does is it allows now commercial entities, which also help to feed life back on Earth, to understand how space helps health or industry or commercialization on Earth to go. And they're all part of our group. So really, commercialization is a part of how we are and how we do our science, just as much as other groups are in terms of funding our scientific research endeavors. It really brings two groups together, commercial and academic research.
And that's something that ASGSR helps facilitate.
Yes, by bringing the communities together and letting our community of academic research scientists understand where the potential is for them to fly. Really helps. And there's only one other place that occurs, and that's the ISS R&D, or Research and Development Conference. But this is really based on the science we do to allow and how commercial can help enable the research that we can do.
I don't know if you've hired a media consultant this year, but everybody's on message with the idea of bringing everybody together under the same roof to talk together. So I'm assuming that that's actually just what goes on here.
Yes, very much so. I mean, we do have our scientists. They, as researchers, right, they go to other conferences, but it's really, the full spectrum of research, from just flat out academic or whatever research that's conducted on Earth without any component to space, to some space work, right? Because they're all researchers, they all have a broad spectrum of science use. This one really is specific to how does space enable research and how is research enabled by space. And that's what's really great. And now you get to talk to a lot of people who do the work, and so collaboration, partnerships, and other things can develop. And what we're also seeing is that scientists and other groups who are potentially non-traditional to the spaceflight environment are starting to come into this community and learning, hey, I have certain questions I want to ask, but I cannot ask them in the environment that I'm in, or I need to look at other extreme environments, and space may provide it. So they come here and they learn more about what we do, and that translates into an increase in the scientific community. Now, the other important factor of the ASGSR is that we are heavily involved in the training and inspiration of students, graduate students, undergraduates, high school students, and middle school students and lower. They're all invited, they all come, and they have an opportunity to learn from our professional scientists, as well as to meet and build networks within each other, because at some point they're all going to be their peers conducting research at a higher level.
Yeah, and I would just say for my part, I was incredibly impressed by the student poster presentations and sessions. I was talking to high school students who are doing work that is well over my head as a as a 40-year-old guy, albeit not a scientist, but there's just some incredible discussions going on at this meeting.
Oh yeah, I mean it's absolutely astounding, and what the ISS, the International Space Station, has done to really drive forward our science and technology and engineering education, our STEM education, even to a certain extent STEAM with the arts.
Yeah.
That would not have been possible without the International Space Station. It's really opened the door to everyone, all the way down to the high school, even to a certain extent the elementary school level. And what's great is the high school students come in and as we become professional scientists, we know boundaries. We know certain things we can and cannot do. These students don't, and it's really refreshing to see these broad ideas and questions that they ask. And I tell you, from the first ASGSR we had them come in, when the students come in, there's just a ratchet increase of energy within the whole ASGSR community when they come through, and I've seen, our scientists just get really rejuvenated talking to these students.
Yeah.
And the students are just absolutely, fantastic. In fact, one year a student said, I wasn't quite sure whether I wanted to go in science, but now that he came to this meeting, he said, this is exactly what I want to do. I want to be a scientist.
Yeah, and I have to say that doesn't surprise me one bit. It's a palpable feeling of energy. When those sessions are going on.
Yeah, and the important part is the students come in, but they don't know that they're doing things, and they have a question. Am I doing something that's real? Is it real world? Right? Because that's always their question. Am what I'm doing really relevant?
Yeah.
They come to this meeting, and they talk to scientists, and they have the realization that, hey, what I am doing at my level to define my experiment, to run our experiments, and all sorts of questions I'm asking, it's the same that the professionals do. So I'm really doing something that is relevant to my future. It's not just something that's just out there. They learn that I am doing something that the professionals do, but at my level. And then they learn and they build from that.
Yeah, it was fascinating. Some of the students I talked to even have an experiment that's, planning to be performed on the ISS in this coming thing, and that's an opportunity that is just incredible for someone of that.
Oh,
age.
it is. It's a wonderful facility, that's really opened the doors to everyone, and from the schools that have programs to programs that NASA has to programs that the ISS National Lab has to help promote student education, student research, whether it's on the ground to train them, or whether it's actually through Suborbital, through Blue Origin and other groups, or through the International Space Station. It's really astounding.
Yeah, has that been a priority for you during your presidency?
Yes, it really has. I mean, overall, it's really been how do we grow and build our community, and how do we show the cross links between physical scientists, fundamental physics, and life sciences, as well as promote them individually. But it's really been a priority also to really build our student community, because in reality, they're our future.
Absolutely.
And so if we can't train them, if we can't inspire them to do wherever they go in the future, we're never going to grow. And so it's really been a priority to make sure we have a strong student foundation here in the society.
That's fantastic. It really does come through. Let's talk a little bit about the meeting. I think we're heading in toward the grand finale. It's getting a little bit late on a Saturday afternoon. Actually, we're going into the awards banquet. Can you tell us a little bit about the Fellows Awards?
So the Fellow Awards, this is the first year we're awarding it, and it's really to recognize individuals who in the society have contributed to the society itself, but more so in terms of their contribution to the science within their field, or their contributions to promoting spaceflight research, to executing in space, and also their contributions to education. So it's really to recognize those individuals who have put forward that extra effort and capabilities to really bring about this revolution and promotion of space life science and physical sciences and fundamental research in space.
Well, thank you very much. And, I'm certainly looking forward to the banquet and the ceremony. It should be great fun. Dr. Sato, thank you very much for joining me.
Well, thank you. I really appreciate the chance to talk with you.
And next up, I had the pleasure of chatting with Dr. Doug Matson of Tufts University, who was also the incoming president of ASGSR at the time that I chatted with him. Thank you very much for joining me, Dr. Matson.
Thank you for having me here today.
Okay, so thought we might first talk a little bit about ASGSR itself. You're the president.
That's correct.
Kind of what are the plans or expectations for the organization for the next year, roughly?
Okay, there's two big things for the upcoming year. The first is that this is going to be the 20th year that the station has been on orbit. And so we're going to be celebrating the research accomplishments that we've had through the past 2 decades, continuous operation with man in space. There's now about a quarter of the world's population who are younger than that, and therefore they have never known a time that there wasn't a man in space. And that's an interesting observation to have.
Yeah, that is interesting. I'd never thought about that.
Yeah. And the second thing is we're preparing now for an upcoming decadal survey, which allows us to put input into the National Academy of Sciences for them to assist NASA with figuring out what should NASA be looking at as far as research opportunities and working towards the next mission to Mars, mission to the moon, et cetera, as part of the developing a coherent plan to prioritize the science.
Okay, and just so I understand the mechanics, who carries out the survey?
Survey is done by the national academies. They bring in a bunch of experts from across the nation, and mainly people who are outside of NASA. And they go through and evaluate, what has NASA done in the last 10 years, and what are the needs of NASA based on where we think the agency is going with future missions.
Okay, and so at this point, you're also then, kind of looking at the types of research that might be done as
Correct,
well?
Correct, and prioritizing that research, and also looking a little bit into how much that would cost.
Okay, and can you tell us a little bit, perhaps what we might be seeing on the horizon?
Sure. Well, right now, as you know, we're in a transition period trying to get the first man and the first woman onto the moon since the Apollo era. And there's a lot of things that have to be done in order to make that possible. It's leading towards having a habitable environment on the moon, and then going onward from there to Mars. And so there's a bunch of tasks in radiation and flame stability and dust management that have huge impacts on the ability for people to safely go and live in space for long durations. One of the other things is we would like to be able to do some manufacturing, say of materials, come up with new materials that have better properties than things here on Earth. But also, if you're on a mission and you have a tool and it breaks, you can't bring a bunch of spare tools along. It weighs too much.
Right.
So we have to be able to go and make tools there in situ so that we can do the maintenance and repair operations that are required for a successful and safe mission.
Yeah, that's interesting because at ISS, it's not exactly a commute to get up there.
Right. It's much simpler, much cheaper, and it now is regular operation that's part of, it's not something that's new. And we have all these new challenges when we think about going to the moon. You would think, oh, we've done it before. But that's not, we went there as a on-off type thing. We were going to be there and we were going to leave.
Right.
Now we're thinking about going there and staying. And that's a completely different mission and has a whole bunch of different challenges, and especially with the fact that we don't have a good simulation for 1/6 gravity. You know, we have a great simulation for the transit. We can do experiments on the International Space Station where there's zero gravity, and that's what you're going to have when you're going to and from the moon or to and from Mars. And so we can do a lot of tests on that, but we don't have something that's large enough to do. Well, how do you transfer propellants from one tank to another at 1/6 gravity?
Right.
Very difficult to try to
that's
simulate. Whereas, you know, we can simulate transferring propellant at zero gravity.
Sure.
So there's a lot of issues that we're going to have to come up with a new way of doing science that includes not only this low Earth orbit, but some suborbital flights and other activities that'll allow us to tailor the gravity to figure out what the physical effects are going to be. The same is true in the life sciences. You know, you have a bunch of, we have this twin study where we've had a person in space for a year. But what about a person at 1/6 gravity?
We don't know yet.
We have no clue.
Oh, that's fascinating. Yeah. So this is an entirely new set of challenges for
Exactly.
ASGSR members and researchers to kind of figure
And
out.
NASA has to broaden its portfolio because, you know, they've been concentrating, you know, in zero gravity activities because that's what we can do. It's cheap, it's efficient, but now we are saying, okay, our next mission is colonization activities. And so, how do we develop the science? How do we broaden our portfolio of what we've looked at in order to make it safe for people?
So that's fascinating. So you're really dealing with a situation that's closer to Earth than dealing with zero gravity.
I wouldn't say closer to Earth.
closer.
I would say it's got its new challenges. I mean, you know, your body reacts to microgravity. It's a stress factor.
Sure.
Well, is it linear?
Right.
I mean, it may be that 1/6 gravity is worse than being at zero gravity because your body says, "Oh, I'm okay," and no, I'm not, and you may actually have mood swings that are completely different. So it's a fascinating thing that we have to start putting in new research activities to look at.
And that's some of the stuff that we've been hearing about at this meeting today.
That's correct.
Why don't you tell me a little bit about your research and what you've presented at this meeting?
So my research activity is looking at the effects of convection on alloy solidification, and it has applications both here on Earth and in space. On Earth, we're looking at optimizing production routes to make better materials with higher reliability, cheaper and faster. But in space, we need to understand just what is the effect of convection on the strength of a part that you would make if you were doing on-orbit repair. So what we're trying to look at is how do metals solidify and where do defect structures develop and how can we control those defect structures so they're not in a place that's detrimental to the part's use.
Okay, so this would be like if I were heating up a piece of metal in order to...
No, it's more in terms of actually making the part from scratch.
Okay.
So when you were doing the first, you start out with raw materials and you're going to come up with a part that's then useful for, let's say you needed a wrench of a certain size. Well, we could make that wrench, and it's actually that process of going from raw material to finished product. It's not trying to look at how one would heat treat it to optimize the performance.
So in that case, then the effects of gravity or the lack thereof has some effect on the way that the metal behaves.
It's actually so when I do my science, I am actually using microgravity as a tool. It's not that how do metals solidify in zero gravity. It's how can I process the material differently than here on Earth. And what I do is, on Earth, I can process the material with no convection or with a lot of convection, but I can't access that region between. In space, I can access that entire spectrum from no stirring to a lot of stirring, and therefore I can understand the underlying science back behind what's going on, so I understand how I can control the part's structure.
And you do see different results.
On Earth, when we have no convection, we see that we get, very nice microstructures, but it's very hard to actually produce those structures for an industrial process. When we go and have lots of stirring, that would be like pouring metal into a casting. And that's very common in industry here on ground.
Right.
By going to space, I look at that continuum between. And so I go to space in order to control the convection. I don't go to space to get microgravity. So it's a subtle difference. It's many operations. If you're in plant science, you want to understand, how do plants react in zero gravity?
Right.
I'm not trying to find out how metals react in zero gravity. That's not what I'm doing.
Okay.
I'm using space as a tool to help me access new regimes of flow that I can't do here on Earth.
And why aren't they accessible on Earth?
The biggest piece of equipment that we use is something called an electromagnetic levitator, and it causes stirring in the sample. And you have a magnetic field that creates fluid flow within the conductive sample. And the weight of gravity pulls the sample down into the field. That means there's a lot of forces required, and there's a lot of stirring. In zero gravity, we don't have to worry about the sample being pulled down into the field. And therefore, we can turn the power up and down to change convection instead of being forced to use the power to keep the sample positioned properly.
Okay, I get it. So this is a situation in which you're able to carry out sort of a mid-level amount of stirring that
Correct.
you couldn't accomplish on Earth
Because you don't have to overcome gravity.
And that has implications for the way that one would build tools in space, which then you might need them as you were going on longer missions and.
Think of it in terms of you've done DIY projects at home and you've got a tool and you break it.
Yeah, I've done that.
Yeah, of course. And so what do? Well, you go to the hardware store and get a new blade. Well, you can't do that in space.
Yeah, you got to make one.
Yeah, you got to make one because you can't bring a whole bunch of spares along because think about your tool set with all the ratchet wrenches in there. Or even all the different sizes of Allen wrenches. You don't want to bring 6 of those.
No.
You just bring one, and if you break one, you make a new one. Or better yet, you don't bring any and make the one you need.
Right. That sounds like an ideal, but it also sounds like it would be a little nervous in case you need that Allen wrench. A last question. Do you have any broad thoughts on the meeting itself and kind of what this brings for researchers and what they're able to do and talk about and get together?
There's several facets to that. The first is one of the advantages to our society is we bring physical scientists and biological scientists together.
Yeah.
And so therefore, when you're listening to a talk in something that's outside of your field, you actually can get these translational ideas like, oh, well, that's an interesting concept. Maybe I can start thinking about how that impacts what I'm doing. The other is, of course, the outreach activity. We have a high component here where we bring in high school students and, of course, the undergraduates and graduate students and give them an opportunity to show the science that they've worked on and to speak with scientists who are doing these activities and give the kids a chance to develop ideas on how to mature what they're doing and make STEM. Actually, I guess the new word is STEAM, which is science, technology, engineering, arts, and mathematics. Which, so to make STEAM real to the students, it's not just some concept that their teachers are telling you. And then the third aspect, of course, is technical networking. I get a chance to talk to my colleagues and meet new colleagues in other disciplines. So I get to talk to people from NASA's Mission Science Directorate that I never would have met.
Right.
I mean, in my role, I work with NASA Marshall.
Sure.
And so I would go to Huntsville, Alabama, and work with the NASA engineers there. But it's a small community, and we're trying to be efficient. But this way, I get to talk to people who are making policy decisions, and policy is not something I normally think about.
Right.
And it's very important because it's leading to the future. Where is NASA going, and how can I be a part of that?
Right. So an opportunity to break down silos and talk to people
Exactly.
that you wouldn't otherwise get to communicate with. All right. Well, that's fantastic. I think I'm going to head downstairs and talk to some of those students. Thank you very much for joining me.
Thank you.
All right, and after my chat with Dr. Matson, I made good on that promise to go downstairs and speak to students. First up was Phoebe Wall, who is an undergraduate at Stanford University, but she was describing some work she did on fluid physics before she went to college, and this work was done in conjunction with Portland State University. Let's go to that conversation. All right, I'm here with Phoebe Wall, who is actually an undergraduate student, and she's here to tell us about capillary action. Thank you for joining us, Phoebe.
Thank you for having me.
If you could just tell us a little bit, why is capillary action relevant for spaceflight?
Yes. So first let's talk about what capillary action is. So have you ever had a glass of water and you put a straw in this glass of water, and you can see the liquid in your straw is higher up than the liquid in the rest of your cup. And so this is because of a phenomenon known as capillary action, in which is liquids have the tendency to be attracted to the surfaces around them. And so the liquid in your glass of water, your water is traveling up your straw because the liquid is attracted to the surface of the straw. So the only reason why your liquid isn't going all the way up the straw is that gravity is pushing it downwards, right? So in the microgravity environment of space, capillary action becomes the dominant phenomenon governing the behavior of the liquid. So it's really important to study capillary action to understand how any liquid works in space in microgravity. And so the systems in space that use liquids might be heat transfer systems, propellant tanks, even life support systems.
Okay, and why don't you tell me a little bit about the research that you just told me about?
Yes. So I am studying specifically adding features to channels where liquid is traveling passively by capillary action. And so I am studying whether or not these features produce a bubble. So it would be interesting to add these sorts of features to a channel because it could improve, for example, the efficiency of a heat transfer system. It can improve the rate of heat transfer by increasing the surface area. But it runs the risk of producing a bubble. And bubbles are bad for spacecraft systems. A bubble can shut down an entire system. So I found a way to mathematically show whether a system will or will not produce a bubble, which allows engineers to design systems they know more definitely will not produce bubbles.
Okay, and these are systems like, forgive my ignorance, but I'm thinking here of heat transfer systems being things like pipes that are sending around a liquid that heats the International Space Station?
Exactly. So one example would be actually heat pipes are an
Okay.
example of a heat transfer system. And these are of particular interest because they use capillary action to transport the coolant. So a coolant will travel through the heat pipe by capillary action along the edges of the pipe, right, reach the end, then evaporate and travel to the other end of the pipe, and then condense back into a liquid and travel by capillary action again. And so it's this time that it's in a liquid phase and is absorbing the heat from the surrounding environment. That's the phase that I'm interested in increasing the rate of heat transfer.
Okay, and so the name of the game here is to increase the capillary action as a means of improving the flow in the system?
In a way, actually adding the features could decrease the flow rate of the capillary action. But my experiment last year that I presented showed that in some situations, if the triangles are a certain size, the impact of the decrease in the flow rate is not big enough to outweigh the benefit that you get from the increase in surface area.
Okay, and this increase in surface area is achieved by what? Is it like the triangle shapes you're talking about? Are they like poking off the side of the pipe?
Yeah, exactly. They also could be a way to model surface roughness. So even if we're not manufacturing these perfect triangles into a heat pipe, just changing materials or changing the roughness properties of the materials can achieve this as well.
And at the same time doing that while not producing a bunch of bubbles that mess up the system.
Exactly, and that's what my experiment is showing this year, is showing when it does or does not produce bubbles.
Okay, and you mathematically modeled this?
Yes, so I created a regime map. So a regime map is basically a graph, and you look at it, and there's different data points in different colors, and each color corresponds to a regime. So now a regime is just a situation that could occur. So one regime is no bubbles are produced, another regime is that bubbles are produced and they're expelled out of the cavity and into the rest of the liquid, or maybe they're trapped by the cavity. And so in a regime map, you can see that the data is separated based on the variables that you have on your x-axis and your y-axis. And then you can draw lines or curves between those different colored areas. And so then in the future, an engineer could look at that regime map and know theoretically where their data point would fall on that regime map and see what colored region it's in. And that color region corresponds to the situation that will occur.
Okay, and then you can use those regime maps to help guide engineers in designing systems that will work optimally?
Exactly, because right now, if you don't have a mathematical model that can show whether or not a bubble is formed, engineers might be really hesitant to add these sorts of channel features to their spacecraft systems, right? So the proposition I made last year of modifying the International Space Station's heat pipes would be risky without knowing whether or not that's going to produce a bubble. And so having a way to mathematically show this allows engineers to take these design changes, not have them be risks to the function of the system.
And something, I'm wondering, and I'm sure our listeners will as well. What's the path to getting here to this meeting as an undergraduate? When did you do all this research?
Yes, so I actually did this research in high school. Like I said, I'm a freshman, so I did this towards the end of my senior year. And I originally got into this source of research through the Celery program. It's a NASA program. It's a collaboration between NASA, Glenn, and Portland State University. And so the Celery Challenge gives high schoolers the ability to conduct capillary action and microgravity experiments. And so it was originally run by engineers at NASA Glenn, and then the drop tower used to get the microgravity environment is at Portland State University.
Mm-hmm. How tall is that drop tower, by the way?
It's about 7 stories.
Oh, that's cool. So you can drop things down the drop tower and simulate a microgravity environment and do your experiments.
Exactly. So you get 2.1 seconds of microgravity. And drop towers are a really great resource because it's pretty expensive to send something to the International Space Station or even in a suborbital flight. But drop towers like the one at Portland State University can be run very cheaply. And so you can do over 100 drops in a day if you wanted to.
Oh, that is so cool. And I will certainly look forward to seeing those designs in the space station in the future.
Yes. And so Portland State University's drop tower is also really exciting because the professor working with it is Dr. Mark Weislogel, and he has done amazing advancements in the behavior of liquids in microgravity. And so I was really honored to work with him on this project.
Oh, thank you very much. And, Mark Weislogel was a former guest on the BioScience Talks podcast. I think I chatted with him last year.
Oh, really?
Yeah, it was great.
That's great.
But Phoebe, thank you very much for joining me.
Thank you so much for having me.
All right, and next up I had the good fortune to chat with 2 graduate students who had just concluded a presentation on biofilms. All right, thank you both very much for joining me. Why don't you go ahead and introduce yourselves, and then let's go ahead and talk about your presentation a little bit.
So I'm Rylee Schauer. I'm a graduate student at CU Boulder, and I also work at BioServe Space Technologies, which is a research group through there.
And I'm Pamela Flores, and I am from CU Boulder as well, but from MCDB department, and I work in BioServe too.
Well, I was just at the talk, and I'm interested to hear more about biofilms in space, the surfaces they grow on, and probably more importantly, the surfaces they don't grow on. Mm-hmm. What can you tell us?
So that's part of our investigation. We're
Sure.
looking at different materials that are aerospace relevant or medically relevant. But the thing about biofilms is that they're everywhere.
Right.
And so that's sort of the problem, is that we need to mitigate these or find a way to countermeasure these through different materials. And so that's part of the Space Biofilms project, which just launched on NG12 earlier this month. And it has 2 parts, a fungal half and a bacterial half. I worked on the fungal half, Pamela did the bacterial half. And we are studying how biofilms form in microgravity, and we have ground controls to compare them against to see what the microgravity and radiation environment does to biofilms.
And I'm going to guess the answer to that is not easy. You can't just say they grow really big or they don't.
So there isn't any data really on the fungal biofilms, but the bacterial biofilms, there is some precedence for spaceflight biofilms.
Yeah.
And they grow a different morphology, so a different shape. There's a column and canopy structure that's been reported, and so we're going to try and validate that and see if we see that again.
Okay.
So there's a little bit of data.
And there's also changes in gene expression that are driving this modified structures that they acquire in space. So that's our one of our aims. We want to know what's behind the changes in structure and what's making them be more virulent or what's making them grow faster.
Okay, and I'm just wondering, why do we worry so much about biofilms in the microgravity environment, on the ISS? Is it of particular concern there? Obviously, it's of some concern here on Earth, but not quite the urgency.
Okay, so right now, the pressing urgence is that we are thinking, and by we I mean NASA, about Mars missions and also long and deep space missions.
Right.
And for those kind of missions, you don't have a scenario where you can send back astronauts to Earth in a medical emergency. And the problem is that the biofilms, if they get pathogenic and they don't get to be treated and eliminated, then they can cause severe infections. And if you don't have the correct way to control that, then your mission's doomed, because at halfway through, you're astronauts are going to be dying, or they're just going to have to come back, and that's not viable for the future that NASA wants or is visualizing right now.
So it's bad news to get sick anywhere, but it's really bad news to get sick when you are on a spaceship that's nowhere near Earth.
Yeah, exactly. And it's also a hazard for the hardware.
Okay.
So, bacteria can secrete things that can biocorrode, biodegrade. Same thing with fungus. And they've been reported on all different components of spacecraft, and they can really hinder the performance of things that you've engineered.
Okay, cool. So there's an engineering cause as well as a human interest one.
Yeah.
So how do you combat them?
Well, we have antibiotic treatment.
Sure.
But when they get resistant, then we need to look for some other therapies. So for our project, we visualize that in the future, if we know the pathways and the genes that are being activated, allowing for these biofilms to go more virulent, then we're going to know specific targets that then we can set our minds to develop a new molecule that will hinder those targets. And then that's like another way of creating new medicines that could treat the biofilms. Also, there's like mechanic ways that you can take out the biofilm from where it's growing, but that's only for hardware, because you can't start scratching in your urethra or in other places in the human. Yeah.
The best way is brush your teeth.
Okay.
Yeah.
Got it. Yeah. Do those count as biofilms?
That's the perfect example of a biofilm. You need to. The thing about a biofilm is that it adheres to things.
Sure.
And so if you don't mechanically disrupt it by brushing your teeth, if you just swish toothpaste in your mouth, that is going to make your dentist really angry.
Oh, okay. Wow.
So you need that mechanical disruption.
Oh, that's interesting. So the friction is a necessary part of it.
Absolutely.
In removing a biofilm.
Same idea for washing your hands.
Okay. Cool.
Yeah.
And how are these experiments actually conducted?
Yeah. So you need specific hardware to launch something to space, because a test tube does not work the same way that it on Earth as it would in space, because you don't have gravity holding your fluid down to the bottom. And so there's very specifically engineered hardware to make sure that you have all the safety levels of containment, to keep the astronauts safe and to keep your science clean.
Oh, that's interesting.
And we keep ground controls that are the exact replica, and we do them. Like in this experiment, we're doing the controls 2-hour delayed, but simultaneously keeping the same times of incubation, the same times where they were taken out of the incubator and then terminated, and for cold storage as well. So we have something to compare to when the samples come back.
Oh, that's fascinating research. So what's next on the agenda?
Our experiment got launched on November 2nd.
Oh, that's cool.
And it already got activated by the astronauts, so right now we are just waiting for them to come back to us.
So you're awaiting data, and eventually this will be published. And
Yeah, so it's on orbit right now, and we are waiting to get our samples back so that we can analyze them.
That's really cool. All right, well, thank you both very much for joining me.
Yeah, thanks for having us.
Yeah.
And last but certainly not least, we round out the main interviews with Rob Ferl and Annalisa Paul of the University of Florida, whose voices will be familiar to you from previous ASGSR podcasts. We start out with just Dr. Ferl chatting about the decadal survey, and Dr. Paul joins us just a little bit later. So why don't you tell me a little bit about the decadal? I just talked to Doug Matson, and we chatted about it a little bit, but what is that?
So the decadal study is, I don't know, especially for us as biologists, sort of writ large as a community, we don't normally think along the lines of having guiding documents that present our science to the policymakers and to the administrators that eventually enable us to do the science that we want to do. NASA, perhaps uniquely among the funding organizations that we as scientists apply to, engage the National Academies of Science to provide a decadal and every 10-year study that aggregates the information that we've learned in the last few years, in the last decade, and then tries to predict, works at a good sort of science, forward-looking, even modeling perspective to guide the agency in terms of what the important science that is able to be done, that ought to be done, and that really should be done in the next 10 years. So all of the directorates, all of the science mission capable parts of NASA produce decadal studies. They range everything from heliophysics to planetary science to Earth observation science, and now also the science that we do in space for the exploration of space and for understanding the role of gravity in development. And from a biological perspective, it is really the kind of thing that allows us to understand the role of biology in space exploration and how exploration of space teaches us more about biology on Earth. So it's one of the few places where science policy can be put in the hands of the scientists that are looking forward.
That's a really interesting process. So I was talking downstairs with some NASA folks yesterday, and they were talking about the way that they review their proposals. When they do that, are they working alongside these decadal surveys? Are they using them as sort of guideposts and recommendations when they're making those types of decisions?
Absolutely. And I'm going to take just a few minutes to sort of like talk about the evolution of the decadal process, especially over the last 10 years as biology and physical sciences in space has matured in the space station era. Because it really is about the give and take, the learning and teaching, and then the learning and teaching in reverse that enables science to get done on the very large societal scale that we deal with. Regardless of how you think about the purity of science, eventually policymakers in Washington are going to lay the groundwork, lay the funding for our agencies to enable the science that gets done. How the policymakers are informed eventually plays a big part in the science that gets done. Right now, when we are presented with grant proposal opportunities from NASA, there are check boxes on the application where we have to place, identify, and show what parts of the decadal our projects are addressing. So in a very, very real sense, the agency uses the decadal survey as a guiding light for the kinds of science that they're asking for and the kinds of science that they fund.
And just how does that process actually go? Is it a year-long process to conduct the survey? Is it 5 years? Ongoing, continuous? Yeah. Is it competitive?
The decadal studies, the decadal surveys, use the proper term here. The decadal surveys are a large-scale operation put on by the national academies to capture as much fairly distributed input as absolutely possible.
Right.
So you shouldn't feel like you are in competition to get your stuff recognized by the decadal. What you do have to feel like, however, is that if you don't speak up, you might not be heard.
Right.
So the groundwork notion of the conduct of a decadal survey, which takes about 2 years to happen, by the way, is a long series of town halls, calls for white papers, calls for information, calls to visit with the academies and present information, and then the survey is put together by upwards of 100 or more people that represent the community. So you as a scientist shouldn't feel like you're in competition, but you should feel like you better get your stuff in front of that 100 or so people that are going to put together the survey. So there's multiple opportunities. I truly believe that the academies is skilled at conducting these surveys because, as I mentioned, even for NASA, they put upwards of 10 of these decadal surveys together for the various parts and places where NASA does research. So there's ample opportunity to get your science into the mix that is digested, understood, and then predicted forecasted by the group that puts together the decadal survey.
Okay. And this perhaps is an unfair question, but any predictions, prognostications about the types of things that we'll be seeing in the upcoming survey? Yes. So let me wax what I think is gonna be the philosophical sort of forecasting that will be in this decadal that is different than in the past. One is the moon is opened up. Right.
All right. So now all of a sudden, biologists, physical scientists can consider again over the next 10 years what experiments ought to be done on the moon. All right. 10 years ago, that wasn't available. Now that is available. Second big thing is that there are increasingly larger numbers of industry and commercial sector folks, scientists, companies, and investors that are looking to the moon, especially looking to low Earth orbit for commercial opportunities, for science that ought to be done, for example, in low Earth orbit to enhance whatever function that company wants to do. So there are increasingly commercialized interests, more production-oriented science that is likely to be included and likely to be thought about by the people putting together the decadal survey. So going to the moon, increasingly commercialized low Earth orbit, and then also generalized the notion of going beyond low Earth orbit. So what happens in our particular case, what happens to biology outside of the protection of the Van Allen belts? Again, something that we couldn't consider in the past.
Okay, great. Why don't we talk, briefly about some of the research that you've conducted, over the past year since we last talked about it? What kind of things have been on the agenda?
Well, so I've mentioned that the moon is now sort of opened up.
Yeah.
So there's a lot of sort of forward think on what happens at 1,6G and what happens on the moon, what happens in a radiation environment. But what has truly opened up on a very practical basis for science over the last year or so has been suborbital space. And what I mean by suborbital space, I mean the kind of space you can get to in Blue Origin's New Shepard capsule or in Virgin Galactic's Spaceship Two. And so 10 years ago, they were thinking about getting into suborbital space. Now they can get there, and over the past year, a number of scientists have had opportunities to fly experiments into suborbital space. Why is that important? One is that it's easier to get to than the space station. But as biologists, what we find truly intriguing about suborbital space is that we can study the transition from 1G to high G to absence of gravity, and then the reacquisition of gravity in a single experiment. And we can do that in a day.
Oh, that's fascinating. So these ships are, being launched from Earth, and then they're going up into a microgravity environment, or is the effect created by descent?
So that's a truly great question.
Good. I'm afraid it was going to be stupid.
It's a fundamental question, but it really ties together the basic notion that even when you are in orbit around the Earth, you're still in a gravity field.
Right.
It is, however, the fact that you are falling at a constant rate around the Earth that is, in fact, local microgravity. These new suborbital vehicles achieve that for about 3, maybe 4 minutes as they cross a parabolic arc in the high stratosphere and in space, but not achieving the velocities that allow you to fall consistently in orbit around the Earth. So it's very high quality microgravity. It is of a duration that is minutes, whereas other things that we can fly in, parabolic aircraft or even drop towers, can have microgravity for a second or a few seconds. These new vehicles can give us on the order of minutes. So we're a power of 10 more time in microgravity.
Yeah, I remember we talked a year or two ago about the plants' sort of immediate biological responses to being in microgravity for very short periods of time on parabolic flights. So this kind of opens it out another tenfold.
Exactly. And the other thing that it allows us to do is bracket and calibrate what kinds of biological effects are triggered by entry into microgravity or triggered during a rocket ride into space. And it really allows us to understand the flight environment sort of writ in a much larger sense. As opposed to, again, sending seeds to the International Space Station, which we're pretty good at now.
Yeah.
And we're pretty good at growing plants in space. But we don't understand yet how a plant would transition its physiology during a launch or during entry into microgravity. It opens up a whole new realm of experimentation once we understand that, to look at the mechanisms, especially for gravity perception and signal transduction and response to gravity changes.
And what's the interest in that specifically, the launch environment? I understand why we need to know what microgravity does to plant growth, because we would presumably be growing plants on longer missions. Or why do we need to know specifically what happens to them during launch or reentry? What's the
So that's a very fair question, and it really highlights the notion that as plant biologists, we float around in the space world either as people that worry about how to feed astronauts,
right,
or people that try to understand basic biology principles using plants as model organisms. Clearly, in the space exploration business, we're not going to be launching plants to orbit, nor are we going to be asking plants to ride with us down to the surface of the moon. So from a feeding astronauts perspective, we don't really, you're right, we don't care. But if we're looking for places and regimes where we can understand how organisms perceive and transmit signals in response to gravity, now we have a chance to examine those systems where we didn't have that chance before.
Okay, so it's basic understanding of the way
at this point, for suborbital flights especially, it is about understanding basic gravity sensing and response.
Okay, and right now we've just been joined by Dr. Annalisa Paul.
Hi, good morning.
Hi, good morning. So we've been talking a little bit about the kind of science that's enabled by suborbital flight. What sort of research have you been up to since the last time that we've chatted?
Well, in addition to the suborbital work that we've been doing, we're also doing analyses still of some of our last epigenomic experiments that we flew on the space station. And we are gearing up for our next experiment that will be a true epigenetic experiment where we take a look at what happens when plants spend their entire life cycle in microgravity environment, collect their seeds, and then put those seeds back on in another flight, and take a look at how the physiological adaptation matches with the genetic adaptation.
Oh, that's interesting. So is this the first time that seeds have been taken from a plant that's been itself grown on the ISS?
I don't think it's the first time that actually has been done. However, this would be the first example of where we have actively taken a look at what we call the epigenome and the epigenetic effects of how the experience of living and developing in space can essentially imprint the basic genetic mechanisms that then guide how a plant adjusts its physiology to that environment.
Okay, that's fascinating, and we'll look forward to that. Why don't we talk a little bit about the meeting and some of the things that you've been presenting and talking about so far. So any particular highlights, any particular talks that you found exciting or participated in, presentations you gave yourself?
Well, I myself haven't given any presentations this particular time, but it has been a lot of fun. This particular meeting, I think we get better and better about integrating the life and physical sciences to solve the same kind of problems or look at the same kind of questions that we have on what does it take to grow plants or develop animals on a microgravity environment. It takes a combination of both engineering of physical spaces and physical habitats and adjusting how the plants and animals that have to live in those habitats can be chosen, adjusted, engineered.
Yeah. And just as an example, kind of how does that play out in those collaborations between the physical sciences and the life sciences, on the ISS or for planned future missions?
Well, sometimes it's as simple as talking to each other. A lot of times we work in relative vacuums of we look at problems and how things that we need to solve when just down the hall you have somebody who's been thinking about, for instance, how fluid moves in pipes for a very long time, and it's just as simple as getting together and writing things out.
And I should mention, we just heard a talk this morning that basically involved understanding and engineering fluid movement as it would impact interactions with roots. I think this is the first or among the first time when the physical scientists have tried to understand capillary flow in a way that now engenders sort of an engineering approach to being better biologists, better horticulturalists in space. So, for example, they're using sponge-shaped or root-shaped sponges to understand fluid flow, and with the anticipation that once they understand that, they can better engineer their systems to interact with our roots.
And that's interesting. So that's the kind of opportunity that you wouldn't get outside of a venue such as this one.
Exactly. And we're getting more and more proactive about creating those type of interfaces, those kinds of interactions at this meeting particularly.
And how do you go about that? I'm just curious from sort of a mechanical standpoint of how do you encourage those types of collaborations?
Well, sometimes it's very granular, very particular. And so, for instance, when we are designing the meetings for the year, the symposia for the coming year, we have a committee at ASGSR where we get together and think about, well, what would we like to see? And so it's as simple as, okay, let's have a symposium that highlights these type of interactions. And we call up the people who do this kind of work and we ask, would you please come and give us a talk?
Right.
And then we choose also talks from among the abstracts that are submitted, put them all in the same room and let them go.
And I should also mention that the society has, again, as part of the decadal survey that we talked about earlier, encouraged the notion that the administration, that the agencies that we interact with, and in this case in particular NASA, pay attention to those new interactions that are important. So, for example, the driving together of the biological and the physical sciences is a conscious multi-year effort by not only the society, but also the agency and the national academies that made recommendations to this whole group.
Mm-hmm.
Yeah, that's interesting because those types of collaborations, it's not the kind of thing that happens by accident. It takes the kind of concerted effort of.
It's a very conscious effort. You're so right about that.
So one other topic that we could discuss. So we're talking about how do you get people to collaborate? How do you get people to work together? There's another serendipitous kind of thing that I think ASGSR is very good at, and that is bringing people from the outside of our community. And so. One of the things that has been very interesting in this particular meeting is there have been several researchers this is the first time they've come to ASGSR. And they have come specifically because they've been looking at and mining some of the data from microgravity type research and spaceflight research and saying, wow, I see a connection here that's relevant to my work, even though they never considered doing spaceflight research before. So they wanted to come and learn more, and it's been very exciting both to talk to those newcomers, those new people with new ideas and new perspectives, because that really enriches the community in a way that just doing the same old stuff, of course, won't.
Right. And how do you find those people? Are you reaching out to them, or are they reaching out to you as they see the research become or the data become available?
It works both ways. It works both ways.
Yeah. So I assume that all of you have very busy email inboxes.
Yes.
Thank you both very much for joining me.
No, you're very welcome. It was my pleasure.
Yeah, you definitely are welcome, and we appreciate the fact that you're here doing what you do, because in answering some of your questions about how we build communities and broaden our communities, doing what you do is a big part of that.
Thank you. And we'll let those kind words from Dr. Ferl close out our formal interviews from the 2019 ASGSR meeting. But as we've done in the past, I wanted to bring you some additional sights and sounds from the exhibit hall and student presentations. I had a lot of great conversations with students at their displays this year, so please listen along and be sure to register for ASGSR's 2020 meeting, November 4th through 7th in Houston. I'll include a link in the show notes, and thank you very much for listening. Okay, and so are y'all undergraduate students, graduate students?
We're high school students.
You're high school students. All right, so why don't you tell me a little bit about this poster and what I'm looking at right now?
Sure. So for our experiment, we are studying the effect of rest on steel conductivity and microgravity.
So rust is a very dangerous issue. It can degrade the physical, thermal, and electrical properties of metals, and this is bad because we're sending up complicated machinery into space, and we don't want it to get ruined by rust.
Okay, cool. So what have you found so far?
So results are actually pending. We're scheduled to launch February next year.
Okay, great. So you're going to you've conducted the experiment on Earth, and now you're going to conduct it on the ISS?
Yes.
Oh, that's really cool. Do you know when the launch is yet?
It's February 2020, so next year. And it should take about a month, 30 days. So then hopefully it will land mid-March, maybe April.
And then you're going to be here next year with another poster telling us how it went in space.
Yes.
Oh, well, that's so cool, and I'll look forward to hearing from you then.
I was actually part of a summer internship at the Gene Lab for High School summer program, which is in Mountain View, California. And basically, in this program, we were given data sets to analyze, and from there, we proposed a hypothesis and an experiment that goes along with it. My team decided to focus on the soleus, which is a major anti-gravity muscle. And we wanted to see the correlation between that and the insulin signaling pathway. We focused on a gene called early growth response 1, or EGR1.
Okay.
And basically, in the normal insulin signaling pathway, this gene doesn't necessarily play a big role, and insulin signal is able to do its thing, and glucose is able to get taken up into the body. But when EGR1 is overexpressed, it actually binds to the insulin receptor, and that inhibits it and blocks off communication with the rest of the pathway.
So is this a case in which there's a potential maybe risk for type 2 diabetes for those who are in space?
Yes, there is an increased prevalence of precursors of type 2 diabetes.
Thank you very much for sharing that with me.
Thank you.
Yeah, so basically what we're doing is we are trying to find in microgravity if there's a difference between calcium carbonate formation on Earth than in the microgravity. We think that there could be applications with medical research for like osteoporosis medication.
So we're going to be running 2 trials in space. We have a small container, and then once the experiment is in orbit, we're going to combine vinegar and dolomite crystal to form aragonite, which is the calcium carbonate. And then in space, we're running 2 trials, and on ground, we're running 20 parallel tests. And by the time that we retrieve our space samples, we're going to go to the University of Minnesota crystallography lab and compare the crystals to see if the formation, to see how the structures of the crystals are different. It's flying in March.
One
one potential thing that we're looking at is when astronauts are in space, their bones lose bone mass.
Right.
So that's an obvious example. I guess we're trying to look at the formation to see if it's different. But this crystal is also used in treatment of osteoporosis.
Okay.
So we're seeing if maybe the samples that we fly in space to see if those are either better or worse for the treatment. And
How in the world do you wind up doing this as high school students? How does this happen?
Yeah, through a program at our high school mini academy, and I think it's just a great opportunity for us to send a small project with real life applications to help scientists and
Veggie, it's a module for growing plants in space.
That's right. This is the Veggie was developed a number of years ago, but we have two of them now up on the space station, being split between growing plants for crew to eat and for research purposes.
So the astronauts are now actually able to eat the plants that are coming out of the system?
Most of the time. Sometimes the tissue is spoken for by the research people. I mean, right now they're doing kind of a 50-50 split, and it just gives you a variety, which psychologically is important when you're stuck in a small vehicle with somebody for quite a time. So.
What I'm looking at right now are various rotating instruments, pieces of equipment, and so these are able to simulate microgravity?
Yes.
How does that work?
Well, in a nutshell, what they're doing is rotating against the gravity vector.
Okay.
So it effectively averages out over time so that it ends up as zero gravity. If you've ever been on a roller coaster,
I have.
or if you've been on a plane,
right,
or an elevator that drops too quickly, for example.
You got 2 out of 3 today.
You'll experience that momentary sensation of kind of weightlessness or suspension. So what these devices do is capitalize on that momentary weightlessness. So these small platforms that you see here are suitable for small model organisms like microbial specimens that you can grow and culture in the plant seedlings. For example, if we let this go continuously, you can see we've had quite a bit of growth in the plant.
So those are real plants?
Those are real live plants. And if you leave them in there long enough, they will outgrow their containers.
Sure. Cool. And I'm just curious, why is it that everything we see on the ISS in a rack of some sort?
You got to contain it somehow. So,
okay.
This is the way of having everything contained in its place and have all the hookups that you need for power, data, thermal, cooling loops, whatever you need. Communication.
Because if you just put it on a desk, it would float away.
Exactly.
Anything from hardware development to helping people design their experiments to fit into the constraints that is associated with spaceflight. So it's all of it. The BioServe does your ground controls all the way up to getting your results back. So an experiment that just got sent up was a Biofilms in space.
Okay.
So what they were looking at was biofilm formation on different material coupons that were associated with space flight hardware. So they sent up these coupons in test tubes with bacteria and then started the cultures in space, and then they terminate the experiment, bring them down, analyze the genes, analyze the biomass, all of that stuff.
Was that the experiment I was just hearing about?
Exactly.
In the next room? Okay, cool. Yeah, cool. Yeah, that would be kind of a bad thing to find out halfway to Mars. That, all of a sudden your electronics don't work anymore because they're all rusted up.
Exactly.
Okay, cool. Well, thank you very much for talking with us today.
Okay, thank you.